All right, welcome to Friday. We are going to, yesterday, let's start with yesterday. Yesterday you did an activity that you looked at different types of cells. There's a couple major classifications of cells. There's prokaryotic and eukaryotic cells. Bacteria cells are probiotic cells. It's also in the toggle bar. And there's plant cells versus animal cells. So those are kind of our classifications. Starting with like really broad is prokaryotic versus eukaryotic. And then within eukaryotic, we talk about plants and animal cells. Okay, so that's kind of our division. So yesterday, part of the activity was it broke down the root words, so the actual meanings of the words. So pro, meaning before, eu, I think meant true. I think those were the prefixes. I'd have to go back and look. But it talked like before the nucleus or a true nucleus. I think eu means true. Am I correct with that from yesterday?
Yeah.
Eu is true. And it's pronounced EU is pronounced you. Okay, eukaryotic or eukaryotes. And I'll say this multiple times as kind of a memory device that you, Y-O-U, you personally, are a eukaryotic organism. And do your cells have a nucleus or no nucleus? You have a nucleus, okay? So you are a eukaryote, you have a nucleus. All eukaryotic cells are like you, which has a nucleus. The ones that don't are bacteria, or what we often classify as bacteria. There are a few, I think, others, but like around still today are bacteria cells that are probiotic. They don't have a nucleus. So that was one of the things that talked about free-floating DNA. Free-floating meaning it's not bound by a nucleus. It's just floating freely through the cell. That's what that was intended. So today we're going to go through. We're going to kind of use the background information from yesterday's POGL to kind of build upon. This is guided notes, section 1, on cell theory and cell diversity. A couple different ways you can turn this in when we're done. We can do a paper copy. There is a Google Doc that you could make a copy of and you could type in if you so choose, or there is also the PDF version that you could mark up. If you, on Videoland, want a different explanation, there is also this link right here that I go through and I talked over from these slides last year. Or if you want to go through and say, hey, I didn't really catch something, I want to hear that description again, video and video. Also, here are the slides, and here are the slides that we are about to go through. Okay? So, let's talk about the different types of cells.
All right, welcome.
That's me. Okay? So, those are the videos. Someone found the video that I linked, and that was, I don't know if that was yesterday's or last year's or something, but when we talk about a cell, this is what our very first images of a cell kind of look like. And there was a guy by the name of Robert Hooke who came out and he developed a microscope that he looked at cork cells. Okay? So cork is a type of wood commonly used in wine bottles to stop the wine from spilling. There's synthetic cork now, but if we talk like a real wood cork, it's very porous. Used to make bulletin boards out of it. There's some other kind of forms of that. But he was the first guy to actually look at it. And Robert Hooke is the guy who's given credit for naming a cell a cell. Because when he looked at this, he said, boy, those look like a lot of small rooms in a monastery. So where like monks would live, monks or priests would live in very uniform rooms, is kind of how he said it. Okay? So the microscope, I kind of jumped around, but the microscope is one of the biggest inventions in terms of seeing microscopic things. People didn't know a lot about this. It was like an unseen world. And in the 1600s, they started to use the microscope, but Hooke was the first one to kind of look at it and say, hey, these are what we call now cells. okay? And this is a very early design of his microscope. I believe that's Hooke's microscope. Where he has a candle and like a reflector, and then he had, I think it was like a sphere of liquid, of water, that would then kind of magnify, and then another magnifying to like get a nice light. What we now use, pretty common, like, hey, let's use a microscope, 7th grade. You know, those were like unheard of and weren't invented yet. Okay. I'm gonna butcher the I believe he's Dutch. His last name, Antoine van Leeuwenhoek. I don't think it's Lee, it's Van, whatever. But he was the first person to see a living specimen under a microscope. So his microscope looked a little bit different. This is his microscope that he developed, and he looked at pond water. So when Hooke looked at a cell, the cork, he saw dead plant organisms that like had really dried out. So he just saw what was remnants of the cell walls. That's all he saw, cell walls. Where von Leeuwenhoek was able to actually go through, and he was able to see things moving and swimming. Even look at the bacteria that lived between his teeth. Yum. Okay. So his was a little bit different approach. There's a lens right here. He could put a drop of water on the point, and he could screw this up and down to kind of change the height, and he could screw this one to kind of get it back and forth into focus while he was looking at a camera. But he was the first one to see a living organism under a microscope. Good. Okay, so here is a Leeuwenhoek. Hey, you guys on the video. We just watched a YouTube that you're going to have to go up and click in the agenda and watch for yourself about von Leeuwenhoek. The cell theory. See, I'm just going to chop that other part out. Okay, the cell theory is based upon the work of a couple different scientists. Okay, we're not going to get too hung up on the names of these people, but realize it's a combination of work of multiple different scientists over a course of many years. So there was a guy by the name of Schleiden, who in 1838 said that all plants are made up of cells. All plants are made up of cells. Schleiden. Then there was a guy by the name of Schwann, who in 1839 stated that all animals are made up of cells. So all plants, then all animals. And then in 1855, a guy by the name of Virchow concluded that new cells could only be produced from existing cells. And collectively, we kind of call these things the cell theory. We call this the cell theory. Okay? So, there's some components of the cell theory. And you should know what the cell theory is. A theory is an explanation of how or why, remember? So it's an explanation of how or why, not a what. Okay? So it's the how or it's the why. So when we talk about the cell theory, there are 3 parts to the cell theory. And you should know these 3 parts. You maybe want to put a star next to this. This is good study material come time for a test. This is good material to be reviewing when you're doing study guides. This is good material because this is the intent, this is the instruction that we're giving you. This is the content that we are presenting to you because we did not pass out a textbook for you. Okay, we're delivering content in this approach. Okay, so this is a good one. This is something you're going to want to make sure you are aware of. There are 3 parts to the cell theory. The first part is all living things are composed of cells. All living things are composed of cells. If something doesn't have cells, it's not alive. It's one of our characteristics of life that we'll talk about next week. assuming we go outside on that expedition. All living things are made up of cells. Number 2, cells are the basic unit of life. Cells are the basic unit of life, which means there's no living thing smaller than a cell. Cells are the basic unit of life. The smallest living thing is a cell. There are some organisms, I mentioned this I think the other day, that are unicellular, uni meaning one, a unicycle, one wheel. Unicellular meaning it's an entirely living organism that is made up of one and only one cell. And then there are some organisms that are multicellular, multi meaning many, are made up of many cells. And you are made up of many, trillions and trillions of cells. Okay, so cells are the basic unit of life, and all cells come from existing cells. So it's kind of a combination of Schleiden and Schwann, and then Virchow kind of coming up with these 3 things. All cells come from existing cells. There's a process of cell division called mitosis and meiosis, and that's where those other cells arrive from. So we take a skin cell, for example, in our hand, came from the division of another skin cell. It's called mitosis. Okay? All cells come from existing cells. Bacteria reproduce because of cells splitting through the process of mitosis, called asexual reproduction. How can we look at these small things? We have 2 different major tools. There are a couple tools. The light microscope and the electron microscopes are the 2 things that scientists use to really look at this. The light microscope, also referred to sometimes as a compound light microscope, is what we use in our science classes. They're good to see dead or living specimens. They're pretty inexpensive, 100-ish bucks. You could probably get a decent one, and you can go all the way up to like $1,000, but they're relatively cheap. But they have to be thin enough so light can shine through them. You have a light source that has to go up through the lens, up into your eye. So they have to be thin enough that you can shine light through. I can't stick my finger under a light microscope and look at my fingerprints. It's not how a compound light microscope works. There's other types of things called like dissecting scopes. That's a different thing. And then we have these things called electron microscopes. But you can only look at dead stuff, really big magnifications, like million-time magnifications, hundreds of thousands of magnification, I'm pretty sure is what we can get. But they're much more expensive. These are like universities or research facilities would have something like this. High schools do not, because the cost of them are really big. Or just the cost to maintain. Because I think you have to do, like this is the chamber, and there's some special conditions that they gotta maintain, which is just cost to operate is pretty high. There's some really cool imagery though that you can look at. They look at like, they talked about like a bee stinger. You can see like the barbs on the stinger of a bee. You can see all the eyes on a fly. Really cool stuff.
Can you wait a minute?
Hypothetically, if you were to look, if you cut off your finger, could you view it through an electron microscope?
Yes. Yeah.
Not sure why you would want to, but
It was a hypothetical. Yeah, I guess I think the answer would be yeah, you could. So here's some examples of like some of the images. So like this is I searched for a bee stinger. So like that's when you get stung by a bee and you still see the stinger in it. That's why the stinger didn't come back out. It's kind of jagged, kind of like a fish barb that kind of gets stuck and doesn't want to come back out.
Sponge spicules.
What's that?
Sponge spicules.
are you able
Like
to
that's
pull it
what
back
you want
out?
me
That's pretty, yeah.
Sponge spicules. Ooh. Yeah.
So like sponges don't have bones. You know, talking about like in the ocean, right? So they've got part of what gives them form and shape are these little things called spicules. So like these are some pretty really cool shapes, and some of them look like weapons, and some of them look like stuff like right out of science fiction, but that's all electron microscope.
So depending upon, there's two different types of electron microscopes. There's something called scanning, which is more three-dimensional, and there's a transmission, which is more just like, it's a 2D image. Yeah, 2D, and it looks more light microscope.
What's the one in the ball?
This?
Yeah.
That's just
Spicule.
one particular depending on which species, their spicules their spicules are kind of unique to each species, so.
Because it kind of looked like one of the COVID thing.
With the spikes on it. I see. Yeah, I see what you're talking about. So now let's kind of reiterate some of the stuff from yesterday, prokaryotic and eukaryotic cells. So all cells come from existing cells, right? Cells come in a variety of size and shape, and there's this recurring theme called form and function. So the shape, the form of a particular cell performs a specific function, okay? So the shape helps it do its job. That's basically what we're saying. You know, so like muscle cells are long and skinny, and they contract. Okay, a bunch of small muscle cells can contract to make movements in your arms. Okay, red blood cells are kind of donut shaped that they can squash to get through like blood capillaries, the walls of capillaries. Nerve cells can send nerve impulses down these long axons, I believe is the term. The guard cells on a leaf that we'll look at under a microscope open and close to allow evaporation and oxygen to be released. Okay, paramecium had these little oar-like structures called cilia on the outside that kind of allow it to move. Ranges in size all the way from the diameter of one human hair is about, the diameter of one human hair is about 50 micrometers is what this says. That's micrometers. So these cells might be as big as a hair, or like if one hair is 50 micrometers, they could go down to 5 micrometers, so a tenth of the diameter of a hair. I believe I did my power 10 correctly. But all cells have at least these 4 common characteristics. All cells are going to have these 4 things. All cells are going to have a cell membrane, the outermost covering that's going to keep everything in it as a cell. If the cell membrane ruptures and stuff's spilling everywhere, the cell's dead. Okay? So they're going to have a cell membrane. They're going to have DNA. DNA is genetic information. It's the information that tells your cells what to do, how to perform, what to make. They're going to have ribosomes. The ribosomes are very important for assembling proteins, which is something that we need for our bodies, and they actually are what exhibit particular traits. And they have cytoplasm, which is like that gel-like water substance that everything is kind of suspended in. So all cells are going to have these 4 things.
Quick question for you. So from the POGIL yesterday, we saw DNA in both drawings, right? In the prokaryotic and eukaryotic cells. Where was the DNA in the prokaryotic cells? Or where was the DNA located in the prokaryotic versus the eukaryotic cells? Nate,
Nucleus?
for which one?
For the prokaryotic.
The other one.
Eukaryotic.
Because you are a eukaryote and you have a nucleus. So the DNA is contained in the nucleus of a eukaryote.
So in that prokaryotic, I think, and it was described in the POGIL as free floating. It really didn't have a home. It was just kind of suspended in the cytoplasm, right? In the eukaryotic, it was in the DNA. It was in the DNA was in the nucleus.
Yep, it's housed in there. Yeah, it's protected. It's kind of a protection. All cells have those 4 things. And in your prokaryotes, the diagram shows that. So they fall into 2 major categories, kind of depending upon the nucleus. And the nucleus is a large membrane enclosed structure that contains the DNA. Okay, so the nucleus is the defining feature. There's technically we simplify it to say eukaryotes have a nucleus and prokaryotes do not. Officially, it's a eukaryotic has membrane-bound organelles. is the official description of it. Nucleus is the one we kind of go to. So the nucleus has a nuclear membrane, has a membrane that surrounds it that is the nucleus. It forms the outer wall of the nucleus that kind of traps that DNA, or houses, I shouldn't say traps, it contains that. So you are a eukaryote, eukaryotes have a nucleus, prokaryotes don't. The DNA is just kind of free floating, like the Pogo said. So we'll talk a little bit more specifics about each type here. So this is kind of a general comparison of prokaryotes and eukaryotes. But if we say, well, what does a eukaryote specifically have, and what does a eukaryote specifically have, what does a prokaryote specifically have? That's what the last two slides are going to be addressing.
Can you go back one? Alright, I'm almost done.
Sometimes instead of saying eukaryote, we say it's eukaryotic. We change the ending. It's a eukaryotic cell, it's a prokaryotic cell, is kind of how we'd say that. Or we refer to them as eukaryotes or prokaryotes.
Just one more second.
So that's how we can kind of distinguish it. Remember, you have a nucleus, you are a eukaryote, and this is pronounced you. So, prokaryotes generally are much smaller and simpler. They're older, they're more ancient. So when it talks about like pro meaning like before, when we talk about how organisms have changed over time, the prokaryotes were the first. Prokaryotes existed before the nucleus was a thing. Okay, a nucleus came around later in terms of timelines. So they're generally smaller. They do have genetic material. It's just not in the nucleus because there is no nucleus. And that's our distinguishing characteristic of a prokaryotic cell. They still carry out all the other actions that all living things do, and we'll talk about these next week before we go outside. They need to be able to grow. These are like characteristics of life. They need to be able to grow. They need to reproduce. They need to respond to their environment. And they can move. Can move is not a characteristic of life. I'm a little, don't know why that one's listed there, I'll be honest. Okay? Because you can be a living thing but not move. There are some organisms that never move their entire life. So movement is not a characteristic of life. Not sure why that's on my list. We'll address those more specifically next week. Bacteria are the most common examples of a prokaryotic cell that we talk about. So we often think of bacteria that are things that get us sick. Okay? We often think that bacteria are things that give us sick. Realize that there's such things as bad bacteria, they get us sick. But good bacteria is super critical for life. We have, in your body right now, you have, I don't remember the number, lots. We'll just go with lots. Lots of bacteria. And it's necessary. Okay? Your intestines are full of bacteria, which is a good thing. It's good bacteria. Okay? It helps digest your food. It helps release vitamins that you eat, but our body doesn't break down. So we can kill bacteria using things called antibiotics. And if you ever take antibiotics because you have a bacterial infection, the idea is you're trying to kill bacteria, but it's generally pretty, this kind of wipes out all bacteria in your body. So there's sometimes some stomach, we'll just go with stomach issues that are associated when you take antibiotics. So one of the things they encourage you to do is eat some yogurt or take a probiotic, which is going to reintroduce bacteria into your digestive system and help regrow that colony of bacteria because it's good for you and it's helpful. Okay? Otherwise, like I said, you kind of get some tummy issues is one way you can kind of describe it. But bacteria are things that we take antibiotics for. And antibiotic, penicillin, I'm sure you, in some history class, penicillin was the first kind of antibiotic that was used, found in bread mold. And it was because of that penicillin, I don't remember the era it was, but it made a huge impact on survival rate of like soldiers in war. If you were to look up like soldiers like who died in the Civil War, and you were to find a chart of who died of like a gunshot versus died of like a secondary, an infection caused by a gunshot, generally there were more people killed by the infections that occurred afterwards, I'm pretty sure this is accurate, than the actual like got shot by a bullet in the Civil War. Because we didn't have antibiotics to treat that. So come around, I think World War I had antibiotics, I think. I'd have to check my dates on that. It
Do you want me to
made
check?
an impact, okay?
Do you want me to check
Antibiotics
for you?
had made a huge impact in society. So what we get is like, oh, I got a little bacterial infection, take some pills, hey, we're good. Fingers crossed, right? Viruses, and I'm just gonna take a little bit to talk about this. A virus is not the same as a bacteria. A virus is not a prokaryotic cell. It's not a cell at all. Okay, a virus is kind of its own classification. It is not a living thing. When we talk about characteristics of life, we'll address that again, but it's not actually a living thing. So when we talk about like using hand sanitizer can kill coronavirus or COVID, well, COVID's the sickness, coronavirus is the virus. It doesn't actually kill technically because it was never a living thing. Okay, just, it's semantics, I know, kind of thing. But we don't take antibiotics for a viral when we get sick from a virus. So if you get COVID, they don't go, here's some penicillin, this is gonna help take care of the infection or your viral infection, because it's not how that works. It's two separate things. So if you ever go to the doctor and you're like, hey, I'm really sick, you know, I got this like, my throat is sore, I got something going on, they can do some bacterial swabs and they can look at some cultures. And if it's a bacterial, then they can treat it with antibiotics, but they might say, well, we don't know what it is, so it's probably something that's viral, which means
Rest and water and.
let it run its course.
Yep.
Now, there are some antiviral medicines that we have that have been developed that help it run its course quicker. If you get like influenza, there's like Tamiflu, I believe is the name brand, that helps, if you catch it early enough, it can help shorten the duration of some of the symptoms, but it doesn't cure it. Okay, so it's a different treatment. HIV is a different one, is the virus that leads to AIDS, okay? And there's antiviral drugs that can help delay the onset of AIDS, and people can like live, their life expectancy of someone who contracts HIV is now longer than it was back in the 80s because of these antiviral drugs. Okay, so I just want to talk a little bit, there's a difference between a virus and a bacteria, and our treatments are very different, but prokaryotic cells are a bacteria, they're a living thing, where viruses are not, okay? You are a eukaryote, you are a eukaryote. And when we talk about these, we generally talk about plants and animals, but realize there's more to them than just plants and animals. There's fungus, there's protists, there's some other things too, but generally we kind of classify as plants and animals. They're generally bigger, they're generally more complex, they have more stuff in them, they have more what we call organelles inside those cells. The biggest distinguisher is they have a specialized organelle called a nucleus. There's some other ones, like I think mitochondria, prokaryotes, that might be right. I don't think so, because I think that's a membrane-bound organelle. Mitochondria. Anyways, so that the nucleus is the big thing, okay? And in these eukaryotic cells, the nucleus houses the DNA. That's where the DNA is kept, it's protected, it's stored. We generally talk about plants and animals. Common misconception is that a mushroom is a plant. It's not. It's a fungus. You say, but it looks, okay, fine, but it's not. It doesn't photosynthesize. It doesn't go through photosynthesis. It doesn't make its own food, which is one of the characteristics of a plant. it actually eats other things. It decomposes, right? It decomposes dead stuff generally. So because of that, it's not an autotroph, it's a heterotroph. It has to eat other things. So just kind of keep that in the back of your mind. And then there's these things called protists, which are small unicellular organisms that also can make us sick. But there are other things that protists do besides make us sick. But that's why, one of the reasons why if you're out in the woods, you boil your water before you drink it, is bacteria. You want to kill those bacteria so they don't infect you. And there's some protozoans, protists, that can also make you pretty sick, so you boil it to kill those before you ingest it. That's the idea. But there are these microorganisms. Okay, so that's what we're talking about when we talk about prokaryotes and eukaryotes. And then that is that. So prokaryotic cells, you are a eukaryote, you have a nucleus, prokaryotes, eukaryotes, and there's plants and animals. And there are some very unique characteristic differences between a plant cell and an animal cell that we will expect you to know, but we'll get to that down the road. This is something we want you to take pictures of. This is a practice to learn thing. This is something that as of right now, we're going to ask you to submit in Canvas. We might change that down the road. That's a discussion we're starting to have because like we went through this in class. The expectation is you're doing it when we go. So do we really need you to take a picture of it because it's just what we do. It's not necessarily homework. So that's something we're starting to talk about. But for right now, make sure you took a picture of both sides of this and turn it into Canvas so we can go through and grade it. We've been chipping through our to-do list. We'll hopefully have your test scores that we can get back to you hopefully on Monday, but we'll see. We'll see, because we got to talk as a bio group and kind of how we're going to combine scores. So from the short answer and the multiple choice. So we'll kind of move on from there. You have a little, we're right about that time. Make sure you submitted your POGIL activity from yesterday as well if you didn't do that. You only had to do the first 4 pages. Okay, there was a page 5 and page 6, but you did not need to do that, is what we said yesterday. Yeah, have a good weekend, everyone.